So first of all, welcome everyone and thanks for joining this session. So today we'll look at a TYK2 optimization use case where we blend QSAR models, our generic models, and 3D Inside utilizing Makya to drive fast chemistry-guided ideation. So we'll start by looking at the TYK2 receptor, the key interactions in TYK2, and the chemical series we are working with. We will also analyze the best strategy for designing new analogs. Then we will move into defining our generation strategy, meaning how to decide what to optimize and which constraints to set. After that, we'll explore chemistry-guided generation settings live in Makya. And finally, we'll review how we process the data, select the best candidates, and prioritize what's synthesis ready. So before we start today's webinar, this is just a reminder about our webpage. You can scan the QR code or you can click in the link that we will share in the comments. And here you'll find the latest webinars, also different kind of demo use cases, benchmark publications, so you can have a better understanding of what we are doing. So now I propose that we jump directly into the use case of today, which is related to TYK2. So just to start, it's just a quick refresher on TYK2 and why this is an important target. So TYK2 is part of the JAK family where we have JAK1, JAK2, JAK3, and also TYK2. So these proteins sit right under cytokines receptors and act like switches for immune signaling, activating a cascade of pro-inflammatory responses. So if we block this pathway at the JAK level, we can dial down inflammation. And this is why TYK2 has become such a strategic therapeutic target. So one challenge we have with the JAK family is that their catalytic domain, the ATP binding sites, are highly conserved. So if you inhibit one JAK at the ATP site, you often end up inhibiting others as well. So TYK2, however, offers a unique opportunity because located next to the catalytic domain, TYK2 has a pseudokinase domain, which is structurally distinct and also inactive. So this domain can be targeted allosterically, which allowed us to achieve selectivity that is very difficult to obtain at the ATP binding sites. So one of the advantages of targeting TYK2 is that its pseudokinase domain offers structural features that are distinct from other JAKs. So the first key feature is the binding to alanine substitution at position 671. So in TYK2, this alanine creates extra space below the gatekeeper residues, forming what's known as the alanine pockets. So this pocket does not exist in the other JAKs and is one of the main structural elements that enables selectivity. So the second distinguishing feature is at position 687, where TYK2 has a threonine instead of a glutamate, which is found in TYK2, 1 and also 3. So these subtle and meaningful differences are what sets TYK2 apart from the rest of the JAK families and allow the inhibitors to bind selectively to this allosteric domain. So historically, most TYK2 inhibitors were designed to target the ATP sites in the catalytic domain, but we've seen that this strategy leads to selectivity issues. So more recently, focus has shifted to targeting the regulatory domain through allosteric inhibition. So in 2022, FDA approved the first drug of this class from BMS, which is a selective TYK2 pseudokinase domain inhibitor for moderate to severe psoriasis. Here, we're looking at the chemical series starting point of the TYK2 program and the series we'll be exploring in today's use case. So this BMS series was built around targeting the pseudokinase domain. So the lead structure here referred as compound 4 shows sub-nanomolar potency in biochemical assays. So, and the interactions are consistent with high affinity binding. So we see anchoring at the hinge position, stabilization around the P loop, and also engagement near the gatekeeper region. So taking all of these together, this gives us a potent TYK2 binder right from the start. However, a key limitation of compound 4 is selectivity. So it displays activity on JAK1, 2, 3, and TYK2 catalytic domain, which is an idea for a clean TYK2 profile. Additionally, analogs of these lead compounds showed poor permeability as well. So with this in mind, now we break the scaffold into 4 distinct interaction areas. So area 1, the hinge binding region, and area 4, which is solvent exposed, won't be discussed in today's use case. So let's now review in more detail area 2, which is responsible for selectivity issues, and area 3, which contributes to permeability issues. So for selectivity, area 2 is the key pockets. This is where TYK2 differs from JAK1, 2, and 3 critically through the threonine and alanine residues. So our objective here is very simple. We want to explore new interactions in area 2 that exploit these TYK2 specific features. And we will focus on small modification in this region because even tiny changes on the terminal amide can shift selectivity towards TYK2 and reduce JAK off targets. Area 3 is a key driver in the poor permeability found in this series of analogs. So here our goal is to explore area 3 in a controlled and efficient manner to improve ARMI properties. So to do this, we rely on QSAR models for TYK2, PIC50, and permeability built from a clean and harmonized datasets. So this allowed us to evaluate modifications in area 3 only from a 2D perspective while simultaneously optimizing multiple key properties. So let's now move into the design phase and see how we can translate all these previous insights into an AI-driven generation strategy. So in Makya, to explore area 2, we use a fragment growing approach starting from the VMS core. So we guide the generation with a QSAR model trained on TYK2 PIC50 data from patents and medchem literature. And we include a structure-based reward to encourage interactions with the TYK2 specific features such as threonine and alanine. So for area 3, we will again use a fragment growing approach, but here we will guide the generation with 2 models. A QSAR model for TYK2 PIC50 to maintain potency, and a CACO2 model to directly address the permeability issues of this region. So we also complement this analysis using Iktos generic models for rescoring, not as an optimization, and this will help us to quickly filter the results. So now that we have the constraints of our generation strategy, let's look at how we apply this workflow to design better molecules. So the first step is the machine learning models. So Makya allows you to integrate local predictive models as targets to optimize. So these models can predict biological activity or admin properties depending on the data set that you're working with. But these models will contribute in the optimization by providing a quantitative signal to the generators. So now I propose that we switch into the software, and I will explain you how to do the overview of the setups. So also a heads up, in the previous webinar, the webinar 1, we've already tackled different kind of generation settings, 3D structure-based and QSARs from the scratch. So here I won't create them from the beginning. So I invite you to go into webinar 1 if you want to have more information of how to really use them. So let's switch back into Makya. And of course, the first thing that you need to have if you're going to work with QSAR models and if you are in the lead optimization phase, of course, is a data sets. So in this case, I already uploaded one of around 5,600 molecules and with the 3 different targets. So let's take a look at how this data set it looks like. So we have the smiles, and then we have 3 targets, which are PIC50, CACO2, and clearance. So we will build now QSAR models based on these targets. So to do that, we need to go into the module for QSAR here in Makya. And this is also a reminder from previous webinar. In Makya, we work with continuous classification models, meaning that you as a user need to give a threshold in order for Makya to split your data into what is good or what is bad, meaning into what is active or non-active, or for example, permeable or not permeable. And in this way, your molecules will be split, and with this threshold, Makya will be able to train your models. After your models are trained, then you can review and see the results, which is what we're going to do right now. So here we have the results for the three QSARs that we created, which are PIC50, CACO2, and clearance. So for PIC50, we already have the results, for example, the metrics with AUC, recall, F1 score, and also we'll give you the metrics for the confusion matrix. As you can see here, metrics are pretty good, so it means that this model can be used during generations. Then the second model, CACO2, also we have pretty decent metrics as well as confusion matrix. Now the problem arrives with clearance because even if we have a good AUC, now we have a pretty bad recall and F1 score and also a very bad confusion matrix. So the conclusion from here is that not all, this is a good example that not all the QSAR models that you're going to build, they're going to be good because they highly depend, of course, in the data that you're using. And the conclusion is always the same. If the metrics are good, then of course you cannot continue using these machine learning models for further analysis. So going back into the presentation now, as we've seen in the demo, when we look at the performance local models, we see that PIC50 and permeability, they're good to go. However, clearance is a different story because even though the AUC looks fine, the F1 score is very low, which tells us that the model really struggled with recall and or maybe suffered from class imbalance. So since we don't have enough quality data to create the local models with clearance, we'll need to rely on our internal generic ADME models to compensate for this limitation. So in Makya, we have integrated 7 generic models. So these models were trained on a large public datasets and refined through clients' projects. So it gives us a more stable early read on ADME properties across a broad chemical space. So particularly here, we use clearance, and it's also important to clarify that our ADME models are used as rescoring, meaning that we generate molecules with the optimization on QSAR, and then on the top of that, we add generic models to help in the filtering of the generated molecules. So now let's again go back into the software so I can show you how to set up these generic models. So if we go back to Makya now, we need to go to rescoring, and in the rescoring, we need to create a new project. So I'm gonna call it as today webinar demo 3. And when you click next, now Makya will give you a list of all the scores that you can, all the targets that you can use to build your scorers. So in particular here, we have different kind of descriptors, the classical physical-chemical descriptors. We have QSAR, the 3 that we've already created, and also we have the Iktos score. And within the Iktos score, we have our ADME models. As I mentioned before, we have HERG, PPV, log-D, log-S, clearance, permeability AV, and permeability ER. So particularly here, we're going to be working with clearance, but of course, if you want to use all of them, you can do so as well. So now after that, the project is created, and so now we can move into score a new data sets. So here you can draw your molecule, you can select a data set from a data set that you uploaded already in Makya, or you can upload a new one as I'm gonna do here. So here, as you see, we have all the listing of molecules, and then just by clicking save, Makya will be able to rescore your molecules. So this should be fairly fast. So let's check. So yes, the status is already done. So after that, you will be able to open your datasets, and here what you will find is your smalls and then how these molecules scored against the specific targets, in this case, clearance. So here we use different kind of threshold just to see if the molecules are good or bad. Again, these are ADME models. And of course, we use a color code in order to indicate the user if the molecule scores well. If the color code is green, it means that the molecules score well. If it's yellow, it means that the molecule, it is good, but is close to the threshold. So Makya wants the user to have this information. And in this specific case, we don't have any red color code, but if that's the case, it means that the molecule scores bad against your targets. So as you can see, this rescoring with the generic models, it's a very, very good tool if you have data sets or if you have some designs ideas and you want to test preliminary information about your ADME models. So this can greatly help you. So going back into our presentation, we have already seen it. Now, a very important point when using generic models is the applicability domain. So since the global models were built from a very large data set, it's intuitive to think the models may fall outside the applicability domain of your specific project. To ensure that the global clearance models was applicable in this specific series, we compare its prediction with the experimental data. So as you can see in the plot, the x-axis displays the experimental data and the y-axis the clearance score taken from Makya. So we simply rescore the 120 molecules using our generic models and then compare the results with the experimental one. So we assess clearance with the Spearman correlation because ranking is what matters most at this stage. So here the global models show an R of 0.53 across 120 compounds, which confirms that it captures the right trend and has enough ranking power to guide early decisions. So even though our local data wasn't sufficient to train a reliable clearance model, the global model gives us a stable way to incorporate clearance into post-processing. So now let's check our last constraint before jumping into the generation, which is the 3D structural information. So we went into very, very much detail of everything related with this 3D structure base in our first webinar. So I invite you to go there in the link that we share, because there you will find all the settings from scratch. So, but here it's just a reminder. Basically in Makya, we can use the 3D information as optimization. So in Makya, we work with docking, and then also we work with fingerprint interactions that we call contact score. So this approach allowed us to bias the AI towards molecules with those key interactions that you as the user wants to retrain. So once again, let's go into the software so I can show you how this works. So of course, the first thing that you need to do is to upload your docking, your files that you will use for docking. So I already did several of them, and today we will be working with this one. So here, after that you upload your files, you need to choose if you want to work with RDoc or with Bina. Specifically here, we will work with Bina. So after that, we need to set up the contact score, these fingerprint interactions that are important for us. And I already created the settings. And what I wanted to highlight here is that we're going to be targeting an alanine and a threonine 697, because as we've seen at the very beginning, these are the residues driving the selectivity towards the TYK2 only. And so also just a reminder, the way that we work is with a system of weights. So the higher the weights, the higher the importance on these residues will be for the generation. It means that Makya will generate molecules targeting this interaction as the main ones. So now we are ready to jump into the generation, because we already created our QSAR, we have our 3D structure base, and we also have the rescoring ready. So let's go back into Makya, and once again, I won't create the generators. You can check it out in our first webinar, but here we will review two of them. First, the area 3, and then the area 2. So let's go into the area 3 first, I'm sorry. And here again, a reminder. In Makya, we have the chemical, all the chemical constraints, which are the reactive centers, where first you need to upload your initial fragment and from where you're gonna grow molecules. So in this specific case, what we wanted just to remove is all the area 3 and ask Makya to give us diversity around this exit vector. Then for the building block point of view, we ask Makya to work only with number of aromatic rings, maximum of 1, and also a minimum of 1 hydrogen bond donor. From the product perspective, we are working with a molecular weight of 500 as maximum, as a minimum of 0.3 for the QED. And then finally, we have the paints and talks. And these paints and talks lists, we always use them because these are substructures that we absolutely don't want to see in our generated molecules. And the way that Makya will work, it will generate molecules and then will filter all the molecules matching this list. So after that, we need to include the rewards. So in this case, the rewards that we will be using are the two machine learning models that we created, which are PIC50 and CACO2, and we will leave outside the clearance because it was a bad one. And then finally, we're gonna add on the top of all of this our generic models, in this case clearance, and some physical chemical descriptors. So Makya will generate molecules, will maximize these molecules to reach the targets, which are PIC50 and CACO2, and then to all the generated molecules, they're gonna be rescored against the clearance. And so just like this, you are ready to run your generators and to get your results. But before seeing the results, I'm going to just to show you the settings for the second generation, which is the area 2, which is the one targeting the selectivity. So here the exit vector is more or less the same, but now we want to introduce modifications in the aromatic amount. So then, from the point of view of the building blocks, we are using non-aromatic rings at all. And from the point of view of products, we want just small modifications, so no more than 450 molecular weight. And then we have the QED, minimum of 0.3. And of course, again, our paints and talks. We are going to work with QSAR models, only with PIC50 in this case. And then we're going to incorporate the 3D structure base that we've seen together already, targeting the two main residues that are important for us. And just like that, you are ready to run the generators and to see your results. So let's explore some of the results. These are the results from the area 3, so the one with the pool permeability. So here you can see all the listing of molecules, and these molecules, they are going to be sorted by different kind of scores. It can be, for example, clearance, CACO2, confidence, you have PIC50, you also have physical chemical descriptors. And also we've seen in our previous webinar how you can filter molecules based on different targets, and in this way just to select the molecules that matter for you. So particularly here, I want to show you the molecules that more or less we selected because they were good scoring against certain targets. So for example, this molecule, if we click here, we have different kind of metrics, like for example, very importantly, confidence, which is going to indicate the applicability domain of your generated molecules with respect to the QSAR that you're using. Then we have also the two QSARs, PIC50 and CACO2, and we're going to give you the prediction. And then finally, on the top of all of that, we have as a post-process score, the clearance, and we're going to give you, in this case, the results in this molecule is scoring very good against our ADME models. Then the result with respect of the 3D structure base, it's more or less the same, but in this case, we are going to be able to see the protein in 3D because we generate the 3D and then we docked inside of the protein pockets. And very importantly, this molecule is targeting the alanine 671, which was very important for the selectivity. So let's go back into the presentation and just to finish the last parts of the webinar. We already seen all of this. Now that the generator is completed, let's look at how we analyze the results and identifies the most promising candidates. So here are examples of molecules generated from exploration of area 2 and 3. So at the top, we have compounds selected using the 3D structure-based rewards combined with PIC50 models. So these preserve the TYK2 interaction and explore new vectors around area 2. And at the bottom, we combine PIC50 and permeability, clearance, and also similarity rewards. So these focus more on improving ADME properties. However, together, this gives us two complementary sets, one driven by structural information and the other one driven by potency and ADME balance. So these molecules here are examples inspired by the trend that we observed in the previous generator. So they reflect the type of modifications that frequently came up as promising while we were generating in area 2 and 3. So here I'm highlighting designs that incorporate the best elements we saw, improve substituents in area 2 for selectivity, and polarity adjustment in area 3 to help permeability, while still preserving the core TYK2 interaction. So these molecules, they were assessed against QSAR, PIC50, and CACO2, clearance global model, and also redoc. And so in the next demo, I'll show how Makya can reproduce this kind of iterative workflow directly in the platform. So if we go back into the platform here, into Makya, now we can create again the same score as before. I'm gonna name it again, webinar demo 4. And now we are gonna select all the targets that I already mentioned, which are the QSARs with the PIC50, here the QSAR with the PIC50 and CACO2. And then we are gonna be working with the ARMI model, which is the clearance. And finally, since we want to redoc, it's possible to redoc using Makya as well. We are gonna use this molecule. And then as we did before, we are gonna just upload the molecule, the selected molecules that we have. And in this way, Makya will be able to rescore it. So in this case, it will take a bit longer because of course we are redocking, and so this takes a bit more time for getting the results. But I already prepared one that I can show you here. And so basically here what you see is your molecules and then the targets that we had for each one of these molecules. Here, as I said before, I did the rescoring using PIC50, CACO2, clearance as ARMI models, and then the 3D structure base using BINA and contact score. And of course, if you're doing the rescoring, you will be able to see your molecule in 3D in your protein pockets. So with this, you can see that Makya is very powerful, not only for generating molecules, but let's say that you generated molecules in Makya and you got very good ideas, and then these ideas, you can use them as they are, or maybe you can use them as an inspiration to create your own molecules. So in that case, you can use these molecules afterwards and you can rescore them using this tool just to see with different constraints how the targets, how they reach the targets, and if they are good enough against the models that you built in Makya. So now just almost at the end of the webinar. So here what I wanted to show you is the results of one design molecule against the reference ligand from BMS. So here we see the important interactions in the hinge, gatekeeper, and P-loop region. Now, if we compare the initial molecule with the design one, we can see that we are adding additional interaction such as glutamate, arginine, but most importantly, expansion into the alanine pocket, where an interaction with alanine 671 boosts selectivity toward TYK2. All in all, this poses overlay remarkably well with the reference, preserving interactions and adding new ones, showing how Makya's AI-driven generation can suggest diverse structures while maintaining also the critical interactions that you set up at the beginning. So just for finishing and wrapping up this session, once we identify our most promising virtual candidates, the next critical question is, which of these can we actually make? So this is where SPIA comes in, and in the previous webinar, we've already explored how to work with a single molecule. So today I want to show you how to work when you need to synthesize a list of molecules and you want to process them all together. So for that, we have our batch retrosynthesis feature, and we are gonna show you a demo right now. So this is SPIA, and in SPIA, a reminder of our previous webinar, we have all the advanced features, like for example, the maximum number of steps, the maximum price per gram, the delivery time, packaging, all the providers list that we have in SPIA. Then you're able also to work with different name reactions. We have plenty of them that you can play around, and with these name reactions, you can ask SPIA to give you routes with only those name reactions, forbid them, or at least using one of them. And then interactions, which is very powerful because maybe you have a way to synthesize an intermediates, and then you want to ask SPIA to create different routes using these intermediates as starting point. And of course, then you can impose substructures or forbid substructures as well. So here, what I wanted to show you is like, since we already know how to introduce smiles, one smile, now in this case, I want to work with a listing of molecules and not only one by one. So you can achieve that using SPIA. So you can drag your molecules into the software, and then they will be recognized. And then just like that, in this case, we won't play with any advanced parameters. You can start the retrosynthesis analysis of your molecules. So for example, here we have the number of steps for each of the molecules that they were retrosynthesized with the R scores. And then each of these points represent different kind of retrosynthesis routes. And of course, we can see the different kind of disconnections. So if we take a look at one of these molecules, as we've seen in our previous webinar, now SPIA will give different clusters of molecules depending on the disconnection, and then we will have the information on our proprietary retrosynthesis score. So, for example, if we click in the first disconnection and then we click into the first retrosynthesis tool, here we're going to explore all the retrosynthesis tree to synthesize this molecule, and we will have the information of the commercially available building blocks, and then with all the providers, delivery time and price per gram. And then also you can play around with all the references for that specific disconnection. Like for example, here we have the reference for the patents, I apologize, the reference for the patents, and then we're also going to give you like the protocol if it's possible to extract. And also we're going to give you more than one ideas and more than one patents, just for you to have different ideas on how to do it. So going back into the presentation, this was actually the last thing that I wanted to show you about our today's webinar. But the message that I wanted to convey here, that Makya is extremely powerful to generating ideas. But then these ideas, they can be translated in inspirations for your own design. And once you have your own design, you can also use Makya afterwards in the rescoring to see how well these design molecules inspired by Makya, they are reaching the different targets. And so with that, it's all for today's webinar. So thank you very much for your attention, and I will give back the word to Eleonora.